in science have always faced challenges. Nevertheless, despite prejudice and lack of acknowledgement, their efforts have produced groundbreaking scientific breakthroughs. Thanks to these women in STEM, countless lives have been saved by their discoveries today. When we don't believe in ourselves, we should be motivated by these role models. As much as we can, we should mention their names today and tell the tales of how they broke new ground in many scientific domains. Alice Ball. The most well-known contribution of African-American chemist Alice Ball is her inventive work in the early 1900s on creating a successful leprosy therapy. Despite receiving little credit, her achievements have had a lasting impact on medical research. Ball's tenacity and scientific acumen contributed to developing contemporary leprosy therapies, and her legacy continues to motivate other scientists and medical professionals. Birth and education. Alice Ball was born in Honolulu, Hawaii on July 24, 1892. She came from a well-known family. Her mother, a school teacher, stressed the value of education, while her father, James Ball, was a distinguished lawyer. Alice was the first woman and African-American to get a degree in chemistry from the esteemed University of Hawaii, where she excelled academically. She received a scholarship to study at the University of Hawaii because of her academic excellence. And in 1915, she graduated with a master's degree in chemistry. Ball's path into medical research started in 1915 when she became a chemistry professor at the University of Hawaii. Dr. Harry T. Hallman, a well-known doctor at the Honolulu Research Institute, became interested in her at this period. Leprosy, an infectious illness that afflicted the Hawaiian people and was a major global health problem, was the condition that Dr. Hallman was trying to cure. Since leprosy was considered incurable at the time, sufferers endured physical and social suffering as a result of the shame associated with the illness. Ball Method. The Ball Method therapy started when Alice Ball began researching Chelmugra oil, a chemical used to cure leprosy in traditional medicine. Although the oil had some potential, its limited solubility made it challenging to administer naturally. Ball refined the oil to make it more efficient and controllable. Ball created a novel injectable type of Chelmugra oil during her study that the body could absorb more efficiently. Numerous patients' lives were significantly improved by her approach, which became the accepted therapy for leprosy. Ball significantly contributed to medical science with her research on Chelmugra oil. Her creation was the primary cure for leprosy and was extensively used for many years. Alice Ball did not, however, get the credit she deserved during her lifetime, despite her noteworthy contribution. After a brief illness, she unfortunately died on December 31, 1916, at the young age of 24. It wasn't until years later that Ball's contribution to the therapy development was recognized entirely. It was eventually credited to Dr. Hallman, who carried out her work after her death. Legacy. Alice Ball's achievements were gradually uncovered and acknowledged in the years after her passing. Dr. Hallman ultimately gave her credit for creating the therapy in 1922. Ball is hailed today for her contributions to treating leprosy and as a pioneer in medical research. To ensure her legacy endures, the Alice Ball Memorial Scholarship was created to financially assist young scientists from underrepresented backgrounds. Alice Ball's life and achievements serve as an example of strength of willpower and creativity. Her biography is an inspirational reminder of the obstacles African Americans and women have faced in the scientific world, and her groundbreaking research has had a long-lasting effect on the medical community. Despite her brief life, Alice Ball is remembered as one of the most significant individuals in medical history because of her achievements, which continue to enhance lives all around the globe. Gertie Cori. Pioneering scientist Gertie Cori became the first female recipient of the Nobel Prize in Physiology or Medicine. In addition to revolutionizing our knowledge of biochemical processes in the human body, her pioneering work in enzymology cleared the path for future generations of women in science. Gertie Cori broke down boundaries and impacted the scientific world with persistence, intelligence, and uncompromising dedication to her studies. Birth and education. Gertie Elion was born on March 15, 1896 in Prague, a part of Austria-Hungary. Her love of science was apparent from a young age. Despite the restrictions imposed on women in the early 20th century, Gertie excelled intellectually, and her parents, who were both Jewish, embraced education. She followed her love of science and finally moved to the United States after graduating with a medical degree from the German University in Prague. In 1920, she graduated with a Ph.D. in biochemistry from the University of Vienna. Gertie met her future husband, Carl Corey, a fellow scientist, when she was working on her Ph.D. The two developed an intensely cooperative and thought-provoking alliance that would alter the path of scientific history. In the early 1920s, the couple relocated to the U.S., where Gertie worked at several research institutes until enrolling at Washington University in St. Louis. She would conduct the studies that resulted in her work that won her the Nobel Prize there. Corey cycle. It was their most notable accomplishment when the pair started researching the molecular mechanisms behind muscle contraction in the early 1930s. Scientists at the time needed to understand the molecular mechanisms underlying muscle function better. Gertie and Carl Corey's study led them to discover the enzyme chorease, which is essential to the biochemical mechanism by which muscles use glycogen as fuel. Their research clarified the intricate process by which muscle activity is influenced by the breakdown of glycogen into glucose. This finding, called the Corey cycle, greatly expanded the discipline of biochemistry and marked a turning point in the study of cellular metabolism. The Corey cycle describes how the body stores energy in the liver and muscles using glycogen. During exercise, it's converted to glucose, which muscles use as fuel. This knowledge of muscle metabolism profoundly affects several disciplines, such as exercise physiology, nutrition, and medicine. In 1947, Gertie Corey, Carl Corey, and Otto Meyerhoff, an Austrian scientist, received the Nobel Prize in Physiology or Medicine for their outstanding accomplishment. Gertie Corey's honor was particularly revolutionary as the first female recipient of the esteemed prize in this category. At a period when women faced enormous obstacles to academic and professional recognition, her victory was a landmark achievement for women in science. Legacy. Despite her obstacles as a woman in a male-dominated profession, Gertie Corey's perseverance and intelligence were evident. Her accomplishments demonstrated that skill and tenacity could transcend social constraints, inspiring subsequent generations of female scientists. She mentored several young scientists who would make contributions to biochemistry when she was a professor at Washington University and wrote more than 100 scientific articles throughout her tenure. Despite Gertie Corey's death on October 26, 1957, her legacy lives on thanks to her pioneering research and the other scientists she inspired. Numerous advancements in our knowledge of metabolism and muscle function were made possible by her contributions to biochemistry and her discovery of the Corey cycle. In addition to her scientific accomplishments, Gertie Corey is recognized today as a trailblazing woman who dismantled the limitations of her day and demonstrated that women could lead and contribute to science. Rosalind Franklin. The identification of the DNA double helix, one of the most important scientific discoveries of the 20th century, was made possible in large part by the work of English scientist and X-ray crystallographer Rosalind Franklin. Her painstaking study established the foundation for comprehending the structure of DNA, even though her efforts were mostly disregarded during her lifetime. Her legacy has now grown to be crucial to the history of genetics. Franklin's work is an excellent example of women's difficulties in science and the need to appreciate everyone's contributions to advancing knowledge. Birth and education. Rosalind Franklin was born in London, England, on July 25, 1920. She was raised in a Jewish household that valued education and support. After completing St. Paul's Girls' School, she went to Newnham College in Cambridge to get a degree in chemistry, continuing her early passion for science. Because of her academic achievements, Franklin received a Ph.D. in physical chemistry in 1945. She next focused on studying X-ray crystallography, a method for determining a compound's molecular structure. Franklin started her groundbreaking research on DNA at King's College, London in 1951, thanks to her proficiency in X-ray crystallography. Despite experts' efforts worldwide, DNA's structure still needed to be discovered. Maurice Wilkins, Francis Crick, and James Watson were among the several researchers who fought to determine its structure. Nevertheless, Franklin's research produced vital information that would eventually result in the discovery of the double helix. Photograph 51. Franklin and Raymond Gosling, her assistant, took high-resolution pictures of DNA fibers using X-ray crystallography. Photograph 51, the most well-known of these pictures, provided important information on the helical structure of DNA. A key piece of the puzzle was revealed by this photograph and Franklin's meticulous examination of the diffraction patterns. The DNA molecule had a spiral structure. Franklin's research proposed, and eventually proved to be accurate, that DNA comprised two entangled strands. Franklin's contributions were not immediately acknowledged, however. Tensions existed between Franklin and other DNA experts at the time. Without authorization, Maurice Wilkins, also working on DNA at King's College, presented Watson and Crick Franklin's X-ray pictures. The lack of cooperation and respect between Franklin and her co-workers resulted from this betrayal of confidence, and Franklin's autonomous and concentrated study style. In 1953, Watson and Crick developed the concept of DNA's double helix shape using Franklin's findings and other previously conducted studies. This discovery won them the Nobel Prize in Physiology or Medicine in 1962. 4 years before the Nobel Prize was given out, in 1958, Franklin, who had not been mentioned in the acknowledgement, died. Legacy. Although Franklin's contributions to our knowledge of DNA were mostly overlooked during her lifetime, her legacy has recently been re-examined and honored. She's now recognized as a key player in discovering the DNA structure, and many scientists and historians credit her innovative X-ray photos and ideas with helping Watson and Crick's models succeed. Franklin's contributions to crystallography went beyond DNA as well. Her studies cleared the path for developments in molecular biology and genetics, and she made significant contributions to our knowledge of the structure of viruses, especially the tobacco mosaic virus. Despite many obstacles as a woman in a male-dominated sector, Rosalind Franklin's dedication to scientific excellence characterized her life and work. Her narrative inspires scientists throughout the globe, particularly women, and her work continues to be essential to molecular biology. Franklin's legacy is firmly established as a pivotal player in discovering the DNA double helix, reminding us of the need to recognize all contributions to scientific advancement, even if she may not have gotten the acclaim she deserved during her lifetime. Barbara McClintock. American scientist Barbara McClintock transformed genetics by identifying transposable elements or jumping genes. Her groundbreaking research disproved the conventional wisdom that DNA is a static blueprint and revealed the dynamic structure of the genome. McClintock's study drastically changed our knowledge of genetic control and inheritance, which earned her the 1983 Nobel Prize in Physiology or Medicine. Her narrative is one of tenacity, intellectual prowess, and a lifetime quest to discover life's mysteries. Birth and education. Born on June 16, 1902 in Hartford, Connecticut, Barbara McClintock came from a family that valued individualism and scholarly inquiry. She had a strong sense of uniqueness and a fascination with science from an early age. She attended Cornell University after high school, first studying agriculture and then switching to botany. McClintock's interest in cytogenetics, the study of chromosomes and their function in heredity, grew while she was a student at Cornell. After graduating from Cornell with a PhD in botany in 1927, McClintock immediately became a maize or corn genetics research pioneer. She became proficient in sophisticated staining methods and used microscopes to examine chromosomes in previously unheard-of detail. Her subsequent findings were made possible by her early studies on chromosomal crossover, the mechanism by which chromosomes exchange genetic material during cell division. Color patterns and maize kernels. McClintock conducted groundbreaking studies on maize in the 1940s and 1950s, carefully observing how characteristics are passed down through the generations. She noticed that some genes seemed to shift across the genome, upsetting neighboring genes and changing their expression. These genetic components could jump from one place on a chromosome to another. She dubbed them transposable elements. McClintock's results called into question the generally accepted notion that the genome was a fixed and unchanging sequence. According to her theory, transposable elements are essential for gene control and mutation, allowing organisms to adjust to environmental changes. Her research also showed how these factors might activate or mute genes, resulting in differences in characteristics like maize kernel color patterns. The scientific community was skeptical of McClintock despite the importance of her work. She had trouble becoming well-known since her beliefs were considered too extreme for the period. She persisted in her investigation, determined to understand the workings of genetic inheritance. McClintock's discovery was not completely recognized until decades later, when transposable elements were found in other creatures and molecular biology progressed. Her discovery of transposable elements earned her the 1983 Nobel Prize in Physiology or Medicine, making her the first female medal recipient in that area. Legacy. The contributions made by Barbara McClintock go much beyond her identification of jumping genes. Her research laid the groundwork for contemporary genetics and molecular biology by demonstrating the genome's dynamic and adaptable character. Transposable elements are now understood to be essential participants in human health, gene control, and evolution, impacting diseases, including cancer and genetic disorders. The life and work of Barbara McClintock exemplify the spirit of persistence and scientific inquiry. She is regarded as one of the most outstanding scientists in history because she discovered transposable elements which defied accepted wisdom and led to new lines of inquiry. Researchers are encouraged by McClintock's legacy to push the limits of knowledge and bravely and curiously embrace the unknown, especially women in science. Helen Taussig. Many people consider American cardiologist Helen Brooke Taussig to pioneer pediatric cardiology. Her pioneering work in treating congenital cardiac problems, including creating the first effective procedure to cure blue baby syndrome, revolutionized medicine and changed the course of history. Taussig, a pioneer in her area, became one of the most renowned doctors of her day after overcoming major personal and professional obstacles. Birth and education. Born on May 24, 1898 in Cambridge, Massachusetts, Helen Taussig came from a family that placed a high importance on education and intelligence. Her mother, Edith, was a suffragist and an early supporter of women's education, and her father, Frank Taussig, was a well-known economist. Tragic events characterized Helen's early years. At the age of 11, she lost her mother to illness. Despite having severe dyslexia, Helen showed incredible tenacity and excelled academically with the help of her father and tutors. Following graduating from Radcliffe College with a bachelor's degree, Taussig attended Boston University to study medicine before transferring to Johns Hopkins University, where she graduated with a medical degree in 1927. She started her work in pediatric cardiology at the Harriet Lane Home for Invalid Children, a division of Johns Hopkins Hospital, despite encountering prejudice based on gender. Blue baby syndrome. Taussig's most notable accomplishment was her research on cyanotic heart disorders, sometimes called blue baby syndrome. The illness is brought on by congenital heart problems that limit the passage of oxygen in the blood and frequently cause infant mortality. Taussig carefully examined the illness, using her acute observational abilities to comprehend the mechanics behind the oxygen shortage. In 1944, Taussig worked with surgical technician Vivian Thomas and surgeon Alfred Blalock to create the Blalock-Thomas-Taussig shunt. By redirecting blood flow to the lungs, This innovative surgical technique enabled blood oxygenation and reduced blue baby syndrome symptoms. Pediatric cardiology surgery underwent a sea change when a 15-month-old girl had the first successful procedure. Due to the procedure's effectiveness, Taussig and her colleagues gained international prominence, revolutionizing how congenital heart abnormalities are treated. Additionally, it made pediatric cardiology a recognized medical specialty. Legacy. Taussig was a fervent supporter of patient safety in addition to her medical advancements. She was instrumental in stopping the United States from approving the medicine thalidomide, which caused severe birth problems in the 1960s. Her dedication to preserving public health was shown in her testimony before Congress. Taussig received several honors for her service to medicine, including the 1964 Presidential Medal of Freedom. Until her retirement in 1963, she kept up her research and teaching at Johns Hopkins, encouraging many medical professionals to pursue similar careers. Helen Taussig's groundbreaking research changed cardiology and saved the lives of many children. She is a role model for women in science and medicine because of her intelligence and perseverance, which enabled her to overcome significant obstacles. Her inventive contributions to juvenile cardiology and her steadfast commitment to enhancing patient care and expanding medical knowledge are what people remember her for today. The ongoing advancement of pediatric cardiology and the lives of the many patients who have profited from her work bear witness to her legacy. Rachel Carson. The most well-known work of American marine Scientologist, novelist, and environmentalist Rachel Carson is her seminal work, Silent Spring. Her research ignited the contemporary environmental movement and highlighted the risks of unrestrained pesticide usage. Generations have been moved by Carson's love of the natural world, scientific accuracy, and poetic writing to acknowledge the interdependence of all species and the pressing need to preserve the environment. Birth and education. On May 27, 1907, Rachel Louise Carson was born in Springdale, Pennsylvania. She spent her early years traveling the countryside and studying animals since she grew up on a family farm that had a strong connection with the natural world. She wanted to be a writer and developed a passion for reading and writing from a young age. Carson first majored in English when she enrolled at the Pennsylvania College for Women, now Chatham University. However, she kept her love of writing while shifting her concentration to science after being inspired by a biology course. Carson attended Johns Hopkins University for graduate work after receiving her bachelor's degree in biology in 1929. There, she obtained a master's degree in zoology. She was unable to finish her PhD studies due to financial difficulties. Nevertheless, Carson started working for the U.S. Her writing and scientific skills came together at the Bureau of Fisheries, later the Fish and Wildlife Service. Nature writing. Carson's first significant work, Under the Sea Wind in 1941, demonstrated her ability to transform complicated scientific ideas into lyrical and approachable language. She gained global recognition and became one of the most prominent voices in nature, writing with her later books. The Sea Around Us in 1951 and The Edge of the Sea in 1955. These works highlighted the beauty and fragility of the natural environment while delving deeply into marine ecosystems. Her early pieces praised nature, but Silent Spring in 1962 signaled a significant change in emphasis and tone. After becoming alarmed with their widespread usage, Carson carefully recorded the detrimental effects of synthetic pesticides, especially DDT, on ecosystems, animals, and human health. She disclosed how these substances got into the food chain and disastrously affected society and the environment. A wake-up call, Silent Spring questioned the methods used by the chemical and agricultural sectors and increased public awareness of environmental deterioration. Despite strong industrial resistance and personal assaults on her reputation, Carson's claims were backed by solid scientific data. Because of her efforts, public opinion was swayed, leading to stronger pesticide laws and the ultimate outlawing of DDT in the U.S. Legacy. The current environmental movement was also sparked by Silent Spring, which led to the establishment of groups like the Natural Resources Defense Council and the Environmental Defense Fund. Additionally, it cleared the path for the Environmental Protection Agency's creation in 1970. Rachel Carson's impact goes much beyond her books. She exemplified the value of challenging unbridled technological advancement and the effectiveness of scientific communication. Her work emphasized the need for a balanced interaction between people and nature and established the groundwork for environmental research and activism. 2 years after the release of Silent Spring, Carson died in 1964. Although she passed away too soon, her impact lives on. She continues to stand as a testament to her bravery and moral character and a reminder of the enormous influence one voice can have in creating a better, more sustainable future for the Earth. Chu Youyou. Chinese pharmaceutical researcher Chu Youyou created history by discovering artemisinin, a drug that has prevented malaria and saved millions of lives. Chu, who won the 2015 Nobel Prize in Physiology or Medicine, demonstrated the importance of multidisciplinary methods in scientific research by bridging the gap between traditional Chinese medicine and contemporary pharmacology. Her tenacity and inventiveness demonstrate how crucial it is to approach global health issues with commitment and originality. Birth and education. Chu Youyou was raised during social and political turmoil after being born on December 30, 1930, in Ningbo, Zhejiang Province, China. Despite obstacles, she studied pharmacology at Peking University Medical School, now Beijing Medical University, demonstrating a remarkable talent for science. Chu started a career in traditional Chinese medicine and pharmacology after graduating in 1955, concentrating on the nexus between conventional treatments and cutting-edge scientific methods. When the Chinese government launched a covert initiative to fight malaria in the 1960s, during the height of the Vietnam War, Chu's knowledge would eventually be crucial. This endeavor, called Project 523, sought to cure malaria, a disease that was wreaking havoc on both troops and people in tropical areas. Anti-malarial treatments. Chu was in charge of a research team whose job was to find a malaria cure that worked. In pursuit of answers, she went to traditional Chinese medicine, a wealth of old wisdom. After examining ancient writings and traditional cures, her team discovered a potential lead in Artemisia annua, sweet wormwood, a plant that has long been used to treat fevers. After careful testing, Chu found that the plant's active ingredient could kill the malaria parasite. She made a significant discovery when she modified the extraction procedure to maintain the compound's effectiveness. She successfully separated artemisinin, the life-saving medication that would be the foundation of contemporary anti-malarial therapies. Using a low-temperature process based on ancient preparation methods, despite the publication of Chu's research in the 1970s, political and cultural obstacles prevented her contributions from being widely acknowledged outside of China for many years. Her efforts revolutionized the worldwide battle against malaria, and as a result, artemisinin-based combination treatments, or ACTs, have become the gold standard for treatment despite the lack of recognition. Artemisinin-based treatments have significantly decreased the death rates of malaria, a disease that once claimed millions of lives each year. Millions of lives have been saved by artemisinin, according to the World Health Organization, WHO, especially in Southeast Asia and Africa, where malaria is most common. Legacy. The first Chinese scientist to receive the Nobel Prize in Physiology or Medicine was Chu Youyou in 2015. The honor acknowledged her contribution to the discovery of artemisinin and emphasized the value of combining contemporary scientific study with traditional medicine. Chu Youyou's accomplishments testify to the strength of interdisciplinary cooperation and the timeless importance of conventional knowledge in tackling contemporary issues. Her research encourages scientists everywhere to investigate various strategies for resolving global health emergencies. As a medical pioneer, a role model for women in science, and an example of how human ingenuity can overcome insurmountable obstacles, Chu Youyou is honored today. The many lives saved and the continuous worldwide campaign to eliminate malaria testify to her legacy. Jane Goodall. British primatologist and anthropologist Jane Goodall has gained international recognition as an environmental stewardship and animal protection champion. Her groundbreaking work with chimpanzees in the wild has wholly transformed our knowledge of animal behavior, human development, and the interdependence of all living things. Goodall has motivated millions to strive for a more humane and sustainable society through her innovative research, advocacy, and educational initiatives. Birth and education. On April 3, 1934, Jane Goodall was born in London, England. Books like Tarzan of the Apes and The Story of Dr. Dolittle stoked her early interest in animals. She dreamed of living among animals in their natural environment and was especially enthralled by African wildlife. When Goodall visited Kenya in 1957, she met Louis Leakey, a well-known anthropologist and archaeologist. Leakey, who saw her ability and enthusiasm, engaged her as an assistant and subsequently arranged for her to research chimpanzees in Tanzania's Gombe Stream National Park. Even though Goodall didn't have any official scientific background then, her perseverance and acute observational abilities would revolutionize primatology. In 1960, Goodall ventured into unexplored terrain and studied wild chimpanzees in Gombe, where she started her expedition. She used a unique technique, spending lengthy hours closely studying her people and earning the confidence instead of keeping a rigid distance. Her perseverance paid off as she produced several groundbreaking findings that upended preconceived notions about humans and primates. Social behaviors among chimpanzees. The fact that chimpanzees use tools, a behavior previously believed to be exclusive to humans, was one of Goodall's most important discoveries. She saw David Greybeard, a chimpanzee, make sticks to get termites out of their burrows. This discovery compelled scientists to redraw the lines separating humans from other animals, proving that our species' inventiveness and problem-solving abilities are not exclusive. Additionally, Goodall recorded sophisticated social behaviors in chimpanzees, such as aggressiveness, cooperation, and emotional attachments. Her research demonstrated the complexity of chimpanzee emotions and intellect, highlighting the evolutionary connections between humans and our closest kin. Goodall's lifelong quest to save chimpanzees and their environment began with this early study. She established the Jane Goodall Institute in 1977, a group devoted to community-based biodiversity preservation, research, and animal protection. The Institute's Roots and Shoots program fosters a new generation of conservationists, which involves global youth in humanitarian and environmental initiatives. Legacy. Speaking at conferences worldwide and bringing attention to the effects of wildlife trafficking, deforestation, and climate change, Goodall has also supported environmental sustainability and animal welfare. Her kind yet impactful speech highlights the value of individual and group efforts in building a better future for the Earth. Science, conservation, and a worldwide awareness have all been profoundly impacted by Jane Goodall's work. By demonstrating the deep parallels between people and animals, she revolutionized the field of primatology research. In addition to her scientific accomplishments, her steadfast commitment to education and conservation has motivated many others to take care of the planet. Goodall is still a representation of resiliency and optimism today. Her legacy serves as a reminder that we can make a difference and is proof of the ability of inquiry, compassion, and willpower to bring about significant change. If you've watched the video till here, that means you've enjoyed the video. Subscribe. Don't forget to turn on the notifications bell icon